So I'm Mark Effingham. I'm the Chief Operating Officer for UK Biobank and here to chair this next session on genetics. So how we're adding value to the UK Biobank resource by harnessing the power of genetics. So we've got an excellent lineup of speakers, and we're going to hear about both research that's been undertaken using the existing genotyping and imputation data already available, but also to hear about both current and future plans to add what can only be described as a tsunami of genetics data that will be added to the resource in the very near future. We've got time allocated at the end, as with the previous session, where we'll invite back the speakers up to the front to take questions. So we'll hold questions until that point. So I'll also pause for a moment and just reflect back, as it was around this time last year that we released the genotyping and imputation data to all of the UK Biobank research community. And I must admit that I was slightly nervous at the time, and I did have some concerns whether the delivery systems that we'd put in place would actually scale to meet the demand when we opened the floodgates. So with support from the European Bioinformatics Institute, we distributed around a petabyte of data in a very short period of time to over 330 different research groups around the world, and did it in a fair and equitable manner that meant researchers could start to work and use the data all at the same time when we fired the starting gun. Fortunately, and the fact that I'm here today suggests this, the provisions that we did make worked very well. But as an overall program, it really was a significant endeavour requiring a large amount of support from the research community, such as the likes of the working groups that support UK Biobank to date, through to inform the early design of the array, processing the sample, so the sample extraction at Biobank of all 500,000 participants, plus the genotyping itself, and then the substantial quality assurance and imputation analyses which came together to allow us to release a dataset of significant scientific importance. I understand that in the early years after UK Biobank was conceived, it was never imagined that a dataset of this nature could be brought together addressing some of the practical and cost challenges to be able to deliver a dataset in the time frame that we did. And here we are only a short time later, where we are standing on a precipice. We're staring out across a landscape where we will soon have the first tranche of exome data made available to UK Biobank researchers early next year through the combined efforts of a consortia led by Regeneron and GSK, but with the intent to extend that further and exome sequence the remaining 450,000 participants by a broader consortium led by Regeneron over the next couple of years. In addition, and with support from government for initial tranche of funding, UK Biobank is embarking on its own vanguard phase of whole genome sequencing, which will be starting shortly, with a view to sequencing 50,000 genomes over the next 18 months. Really, this will be a process where we will learn a lot in terms of how we process at this kind of scale, and that would inform a main phase protocol where we would seek to sequence the remaining 450,000 participants over a subsequent 3-year period. A key part of this will be to run a number of innovation pilots, and one of which will be to explore what the differences are and the relative strengths of different sequencing platforms and technologies, including both short and long read technologies, and how we bring these together to help inform a protocol that will generate data that will allow researchers to unlock parts of the genome previously hidden. I think getting hold of the data is only one part of the challenge. Given the size and complexity, how we actually make those data available to researchers in a way where they can start to use them and obtain the greatest value is a challenge in its own right, and certainly one where we need to be working with the research community over the coming months and years to understand how we address some of those challenges. So it's a very exciting time for Biobank and the research community with what's coming down the line. So to kick this session off, it's my privilege to introduce a short video from Dr. Eric Lander, Professor of Biology at MIT and President and Founding Director of the Broad Institute of Harvard and MIT, with his thoughts on the value of UK Biobank and how adding genetic data at population scale is enabling groundbreaking research.
What's the UK Biobank? It's the thing we always dreamed of. The idea that there would be a huge population collection with information about so many different phenotypes, coupled to genetic information, and that all of that information would be shared freely with bona fide investigators in the simplest, most fluid way. When lots of folks were sequencing the human genome, we imagined that someday in the far future, we thought, it would be possible to learn from whole populations, from the experiences of tens of thousands, hundreds of thousands, someday millions of people, which genetic variants had what effects on people in what settings. Now, disease studies, where you look at people who are already affected with the disease, well, they can teach you a lot. They can teach you what genes play a role, but they don't really give you the whole picture. They don't tell you who doesn't get disease despite having the genetic variants. They don't tell you what environmental factors may play a role as well. And disease studies are usually focused around the limited number of conditions and a limited number of questions. What the UK Biobank has done is made it possible to explore so many different questions, combinations of phenotypes, and to do it with incredibly rich data. It is the model of what we have to do around the world. I think every country is now looking, or should be looking, to what the UK has done as the blueprint for what other countries have to do, so that eventually we can turn our healthcare systems and our populations into incredible learning systems, so that we can guide medicine based on real experience of actual populations, not just clinical trials, not just disease studies, but bringing together broad populations and clinical medicine. So I want to say hats off to the UK Biobank. Everybody will be following your lead. Already all of us are following the data. What you have done is world-changing, and thank you.
So our thanks to Eric Lander for taking the time out to provide what are really kind of inspirational thoughts about what UK Biobank is doing. And I think really frames the context of the sessions we're about to hear. And certainly having watched that video in preparation for this, there are a large number of UK Biobank colleagues here today. I think that just reinforces the reason why we do what we do.